Hi students, this is Marius Pfeiffer, Dr. Marius Pfeiffer. I'm your instructor for the laboratory portion of Biology 1406. Dr. Nithya Joseph is your instructor for the lecture. So I just want to walk over the steps for getting your laboratory started. We're going to look at the menu and Blackboard right here and then go in and actually look at some bits and pieces. I want to call your attention to the announcements that come out. So I already sent you out an announcement asking you to complete the questionnaire. And for those students who have done this, thank you very much. If you have not yet, please finish the questionnaire. This link will take you there. All right. There are a few other announcements down here. This is from Dr. Joseph right here. Follow her instructions for the lecture. And so just to distinguish, Dr. Joseph takes care of your lecture portion, and I take care of your laboratory portion. We've worked together for many semesters, so we know what each of us is doing. You can ask either of us about either bit, but for specific and accurate answers, ask Dr. Joseph about the lecture. Okay. So let's go ahead and take a look at some things. First, my contact information and hours. I have this. Oh, wait a minute. I promised I would do this. Hang on a second. Let me go back. I'm going to switch this over into student view so I see it just like you see it. Okay, there we go. So here we are in the announcements. Let's go ahead and take a look at my contact materials. You can reach me by phone. This is actually my office phone at TCC, but it's forwarded to my phone. Feel free to call. If I can answer, I will. Email, which a lot of y'all have used already, go ahead and use email. That'll be our most regular way of communicating with each other. Be sure to use the mytcc.edu extension right here. All of the faculty have a staff email that's Outlook. It's, Outlook Microsoft email. And it's the one that just has the @tccd. Please don't use that one because we get a huge amount of email on that channel, and it's hard to see students' emails sometimes, and we don't want to miss them. So that's why we have a separate email for y'all. Okay. I also will have regular office hours, which means I'm available and looking for communication from y'all between 9:30 a.m. and 12 noon. You can try and reach me at any other time of the day, but I've got a lot of meetings in the afternoon and other obligations that keep me from being available by phone or email or collaborate, get-togethers or anything like that. Okay, so let's go ahead and take a look at other things we've got right here. I want to point out that the laboratory materials are under this 1406 laboratory materials folder. We've also got this collaborate video meeting space, so I'll just pop that open real quick. And this allows us to have meetings on a regular basis. So here's office hours for Dr. Joseph. These are her office hours. I'll ask her to title them to include her name on them since we share a space right here. And I'll have an office hour during my office hour times right here as well. And then we can also always meet in this course room. And students, y'all can meet in this course room. You can go in here and you can join the course room and you can discuss things amongst yourselves or share information. So you can do the same collaborate sessions that we all do. You have access to this as well. Okay, let's go ahead and take a look at the laboratory materials. So the first thing I want you all to do is, and this is arranged in what are called modules. So if you hear anybody talking about modules, that's just it. It's like a folder with a whole bunch of things inside it, or a binder, so it's a discrete bundle of material, and we've arranged these modules by week. So week 1, week 2, week 3, you can see that. So let's go ahead and take a look at the syllabus. And there's always a checklist at the top right here, and the first one is just read your syllabus. So let's go ahead and do that. And so if you open up your syllabus, you'll see what we're doing each week through the semester. So week 1 is going to be lab safety, introduction, microscope training, etc. You've got things due. They're always due Sunday night at 11:59 p.m. Later in the semester, we're going to be writing a research paper in steps. Those are also due at 11:59 p.m. Sunday night. So everything's due Sunday night, including this week. You got a couple of simple things to do in this first lab this week. All right, and if we scroll on down here, you can take a look at the grading scheme. So the questions that come inside each lesson that we're taking each week, that's 7% of your lab grade. The analyses that you have to do after you've completed the lesson, that's 38% of your lab grade. So that's a big part. Those are really important. And these are the ones that have to be done with the secure proctor, which I'll talk about in a moment. 40% for your lab research paper. You don't have to worry about this for a little bit, but by mid-semester we'll be working on this one here. And then you've got a couple of presentations and a poster to do that get you the remains of your grade. Materials that you need for the lab, well, it's pretty simple. You need a lab notebook. So go ahead and get yourself one of those composition notebooks. I don't have one here. I should have had one ready as a prop, but you've seen them. It's the paper-bound or sometimes plastic-bound small notebook with the black and white speckled cover, and all the paper pages are all sewn into it, so you can't peel out paper without being obvious that you've pulled out a paper. That's an appropriate lab notebook. So get yourself one of those and write down all the notes that we instruct you to do in lessons. And you can use that notebook as your reference guide while you're doing the analyses in your laboratory lessons. That's the one thing you're allowed to take into the analysis with you. All right. There's other information right here, but I think you can read it yourself, and if you've got questions, that's what I'm here for, to answer questions. But go ahead and read over the syllabus. I won't go over the rest of it. Y'all can do that yourself. All right, the third thing in this content is instructions for setting up Proctorio, and it takes you straight to this website right here. And you need to go ahead and, not all the time, but when you're taking the analyses, when you're doing the analyses or for lecture tests, you need to be doing those in Google Chrome, the Chrome extension, the Chrome browser, and you can use this on a PC or a Mac. Chrome is available for both, but Proctorio only works in Google. All right, so in Chrome. Once you have Chrome installed, you need to install the Proctorio extension. That's what this little video here is for, and that's what this little video here is for. It takes just a couple of clicks. It's just a couple of things. As they say, 30 seconds. It's awfully fast. So you'll go ahead and do that, and then you should be ready to go. That's all you need to do to be able to take tests with our proctoring software, which is mandatory for this course. So it is, you've got to get this done. And you want to get it done this week because your first assignment in laboratory requires the proctor due Sunday night. All right, so that's that. Let me close that out right there. And let's go ahead and go to the next module right here. Notice I could navigate back and forth using this sort of nested series of choices up here. So week one, you've got a checklist. These are all the things you need to do, and do them in order. So go ahead and go through the lab safety lesson. Here it's coming up right now, and so you're going to go through here. You've got a bunch of different things. When you get to the end, so there's lab safety information. A lot of this is as if we were having lab face-to-face. It's one of our learning objectives that's required for this course. So even though we're having a virtual lab, please go through this laboratory safety stuff about how to conduct yourself in an actual lab. It's still important. And so I'm just scanning all the way down through here, and I'll go to the next chunk, which is a bit shorter, and you can see how we can move through here. You need to go ahead and do this simulation right here, and when you do this simulation, it's going to take you to an external website. You're going to go through here. You're going to get a certificate when you're done, confirming that you've gone through this training session on laboratory safety. When you get that certificate, save it to your computer because you're going to need it later. All right, so now we're going back to the lesson, and you'll have completed this laboratory safety training film. And then we've got some questions for you to answer. Go ahead and answer these questions right here. You don't need Proctorio for this, only for the analysis that comes later. Okay, and when you're done with this, that's the end of this particular exercise, the takeaway right here. Okay, so we're done with that particular bit. All right, so now let's go on to the next thing, assignment lab safety. Here you're going to hand in that certificate. You'll upload it by browsing your computer wherever you saved it. So make sure you save it. Browse to your computer, find it, and upload it. Okay? Lesson 1. This is a bit bigger. So as you see, we've got microscope use, introduction to eukaryotic cells, scientific method. First, we're going to learn about microscope types. Each one of these lessons has a bunch of objectives. Here's everything that we are expecting you to be able to answer questions about at the end of this lecture, or at the end of this lesson. So here's the objectives. They look a little bit long. The first one's a little long. And then we address all of that material. Microscope types, microscope components. And as you work your way through these pages right here, I probably already skipped some, there's always one or two questions scattered about in here. Please answer these questions. These are graded. You don't need the proctor, these built-in questions that are built into the lessons, but they are the quiz grades, the in-lesson quizzes that are 7% of your lab grade. So it's almost a letter grade just for these guys right here. Which the purpose is to get these right. So read the material, make sure you know the right answer, then, click your answer and put it in there, and you'll get feedback to all of these, all right? So, for instance, the purpose of increasing contrast is to increase magnification. Check answer. That was incorrect, so, you'd want to do it correctly. So keep working on those till you get them right. You have 2 goes at each question. Each one of these questions you can do twice. So if you don't get it the first time, do it again. The best grade of those 2 choices is what we'll take. All right, so carry on through the lesson. So we'll get all the way here to the end, scientific method. There's videos, there's all kinds of stuff. There's lots of different tools in these lessons. We're having to do this online, so we've created a virtual experience for you. And then putting it all together, you've got an assignment right here. All right. So I'm going to ask you in your notebooks, and I'm sorry, I could never make this thing fill in for some reason here, but go ahead in your notebook after you've answered questions about all of your objectives and whatnot. Go ahead and make this table in your notebook. Just recreate it. It doesn't have to be really pretty, but nice enough that I can read it. Nice enough that I can read it, okay? And then you're going to click this link right here to open up images. And so right here, we're going to pull up this picture of Chlamydomonas. And it says Chlamydomonas, that's the genus species. Homo sapiens is our genus species. Homo species would be just, Homo is the genus. There's no other species in our genus these days. They're all extinct. But if there were, Homo would cover all of those. Homo neanderthalis was another species that doesn't exist anymore. Anyway, they're extinct. Chlamydomonas is the genus of these little single-celled organisms, and the magnification in this picture is 400 times. So every one of these pictures has the genus name and the magnification that the image was taken of. And these were taken by our staff at school. This is stuff you would be looking at in lab if we were able to have lab face-to-face. So you're going to go to those pictures, and you're going to use them at least, what is it? What did I say right here? 5 slides. So do at least 5 of those slides. 1, 2, 3, 4, 5. That's why there's 5 rows here. Give the name of the organism. Can you see any structure inside the cells? That's what we mean by organelles. So there was a previous bit on eukaryotic cell biology, so that's where you'll know about these things, eukaryotic cell types. Okay, what color are the cells? And then you're answering a hypothesis. Our hypothesis is that these are eukaryotes and autotrophs. So based on whether they have organelles in here, gives you hypothetical information to assume that they are eukaryotes. If you can see color inside the cells, that usually means that they're autotrophic, that they are photosynthetic, because they must have colorful pigments. Green, red, blue are the typical colors of photosynthetic organisms. And so you're going to complete this table. All right? And that's the last portion of the lesson. All right, putting it all together, and that's the end right here. Let's see, we've got a wrap up. And it says, when you complete this activity, upload a full photograph of the relevant page of your notebook. That's with that table. And you're going to submit that as a file during the post-lab analysis assignment. Okay, so now we're ready for our post-lab analysis. So let's go back. Practice quiz to test Proctorio. you'll want to go ahead and take this practice quiz. It's got 6 questions in it. It will give you a grade in the quiz, but it doesn't go into your grade book. This is just to prove Proctorio works. You want to use it to make sure the Proctor works. Do it as many times as you need to until it's working. Then when you're set, you'll go ahead and do the post-lab analysis. And I'm wondering why it's not showing up right here. Okay, that's a little odd. That's why I was kind of surprised. The post-lab analysis, I've got out of my student view. I'm in my faculty view right here. So for some reason I can't see it when I'm in my student view. So if y'all can't see this post-lab analysis, eukaryotic cells remotely proctored, do please let me know and I'll see if I can make it work. You can see that I've got the Proctorio Secure Proctor installed. This is just for me. Y'all will never be able, should never see that in your laboratory files, but if you see a picture from my Blackboard, that's what this represents. It just shows me that I've got it installed in Blackboard so that I can remotely proctor these analyses right here. All right. So when you do the analysis, it's going to ask you to do a few things. You'll have to check to make sure that your video recorder is working. Here I am on video. That is obligatory. So if you have not got that camera installed yet, let's get it. Okay. Buy a camera. You're going to need it for other classes anyway. If you're working on a laptop or a tablet, you should already have a camera built into it. Get a microphone. I'm talking on headphones with a microphone right here. This works best. You can use speakers and a built-in microphone on a tablet or a notebook, and that will work, but you'll find that headphones, and certainly the microphone associated with headphones, give you the best experience and the people you're talking to the best experience. So I strongly recommend that. You must have a microphone and a video camera to take the analyses and tests for this class, but you've got till Sunday to get that set up. You'll need your ID, so keep your wallet at hand and pull out your ID and show it to the camera when you begin getting ready for the test. So here's my ID. I'm just, here we go, etc. Okay. It may ask you to sweep around the room a little bit, and then you're ready to go. What the Proctorio settings will do is keep you from doing anything else except the analysis assignment on the computer. You won't be able to change pages until you've submitted that analysis. You won't be able to browse. You won't be able to look at other screens, anything like that. Okay, that's obviously necessary. It's going to look for you doing unusual things as long as you're staring, at the keyboard or at the screen and the keyboard and answering questions and looking at your notebook, which I suggest you show me occasionally that you've got your notebook in front of me where you can write down anything you want. Any information you want can be in that notebook, but that's the only resource you can use. Go through the analysis. All right. One of the questions in the analysis is going to ask you to upload a photograph of your notebook. Why don't you go ahead and take that photograph in advance and save it as a file so that you can grab it and upload it into the analysis when the analysis assignment prompts you to do that. All right. So that's how to get going, and that's the beginning of the lab, and that's your first week assignment. Okay. So I'm going to go ahead and close for now and post this. Let's get to work. All right. Our quick reminder, go back to the beginning and go over the syllabus real quickly. Then go into your week 1 module. And let's go ahead and get each one of these done. Lab safety, the lab safety certificate upload. You're going to do the week 1 lesson. Do a practice quiz whenever. It can be before or after the lesson. It doesn't matter, but we put it in an order right here because you've got to get this practice quiz done before you can do the post-lab analysis. All right. And then, yeah, this is just for me. This just shows me that I've got it installed. All right. I'll leave it for now. Email me or phone me if you've got questions. Have a great week. We'll talk to each other soon.